This remains the structure of the thesis. Thesis always begins with as per the university norms, Savitribai Phule University or the Mumbai University, or if you go to even other universities, then always it begins with the introduction, review of literature, material and methods, results and discussion, and the references. This is the structure. So what to write exactly in the introductory part or in the introduction? So in the introduction, cover the background of your topic. Then you have to write about your research problems and the significance of the work. And in the introductory part only gives idea about the structure of thesis, means how many chapters are there, first chapter, second, for introduction, all these chapters. So write about each chapters, then the scope of your topic, and finally objectives. Because number of times student asks what to write exactly in the introduction. Then these are the few points. With the help of this point, you can complete the introductory part or the introduction of your thesis. Then the material and methods, as I said in the beginning only, details of the site or authentication of the material. Always you have to begin this. Then experimental protocols. When you fix the protocols, then here also you have to be very specific. For example, you would like to determine, or if you would like to quantify the amount of protein specifically in crops. And all of you know, or all of you aware with the different methods of protein estimation, generally Lowry's method, Barford method, all these methods are there. And generally we prefer these methods, Biuret method or the Lowry's method. But here, when you go for the referencing, when you do reference, you realize that most of the people, if you go to the various institute, then these people or the most of the researchers, they avoid to use Lowry's method or the Biuret method or the available such kind of methods. Here, when the plant material is crop, you have to go for the Geldal method. Even if you go to the agriculture universities and if you ask about the protein estimation, then these people will tell you about such kind of methods. So specific methods are deals with a specific plant material or specific crop. Then here one more thing I would like to tell you that, for example, Lowry's method. Number of manuals are available, number of books are available. Most of the students, they refer the Sadashivam Manikam. So this book is available. In that book, you will get the protocol of the Lowry's et al. 1951. Okay, so you will get the complete protocol there. But at the end of the protocol, you will find the research paper, you will find the reference of the paper. Nobody read the original paper. So what is important? First of all, read the original research papers of Lowry's done, and then try to understand the given protocol. If you do this, then it will help you a lot. Because original, what exactly, what text, what method, what protocol given in the paper, that is most important. And definitely, when you choose a specific protocol, then why you have chosen a specific protocol, that justification is most important. As I said, Lowry's method or the Jejal method for the crop, various crops, or the even pulsations, because these methods are more suitable. Jejal method is more suitable, more sensitive, specifically for the crops. So justification this justification is most important, and it's possible to justify only when you understand the original protocol. Then one more thing is that when you perform the experiment, again I'll come to the protein only, protein determination of protein. Then while writing the text, while writing the material methods, do not write the protocol as it is, because when you carry out, when you perform the experiment, you have to modify the experiment as per your material. You have to modify the experiment as per your planned material, and uh, the modifications are most important. What exactly the referee or what exactly the committee member expected, they expected means how you exactly perform the experiment, what kind of changes you have made during your experimentation. So even when you write the material method chapter of your thesis, then do not write protocol as it is. Write as per your experimentation, or how you modify the experiment or tackle the issues. So this is most important. Then important thing is that next, when you talk about the specific protocol, whether it is protocol for the protein, again here the, when you talk about the protein or the phenolic compounds or alkaloids, then you have to fix the protocol. And for example, if you would like to go for the hiring services for a specific, even if you would like to work on the proteomics, then here the sample preparation is most important. Even when you go for the experiment in your lab, or if you would like to go for the hiring, then sample preparation is the basic or the most important part, because we focus on other areas and we never given importance to sample preparation. Sample preparation is the fundamental, or we can say the entire result, or the expected result, entire result based on the sample preparation. And you will find various methods of sample preparation. So here also you have to find out suitable method or desirable method, or the method which suit to your plant material. So first of all, you have to focus on the sample preparation. And when you would like to go for the hiring services, then generally these people ask for the sample, and to curtail the expenses, you provide the sample. So do not do like this, because the specific protocols are very specific. Okay, always when you go for the hiring services, then ask that particular agency for the sample also. Means they will prepare the sample and they will do rest of the work. Sample preparation, in variations you will find a gap. If you prepare a sample and send for the hiring, and or these people, institute people, they do the sample, and when they complete the part, you will find the gap in the result. So to avoid these things, always take care of sample preparation. Then the next important point, the use of appropriate tools for data processing. As I said in the beginning only, number of softwares are available, and you have to use this software. And here also remember, number of hiring services are there. They allow you, or they provide you the number of services which can be used for the data processing. Um, if you go through the various research papers, then you will find n number of styles. You will find different graphs. Few graphs are really fantastic, and we thought that how these people go for such kind of graphs. Remember, these things are very simple. It absolutely depend on the software. So try to find out the institutes or the laboratories where you will get these facilities, But for the basic data processing, for example, ANOVA, correlation, regression, okay, similarity and dissimilarity matrix, for all these things, SPSS is more than enough, or even you can go for the Excel or the SPSS. Um, Next, again, I'll emphasize more on the material method because this is the most important part. For example, if you select the plant material, whether it is the plant species or a specific crop, then always how to begin with the research. First of all, obviously, you have to find out the nature of the plant material or the nature of that particular crop. So the most important thing is the understanding of the plant material or understanding a particular crop. Without understanding the crop or the plant material, it's very difficult to carry out the research or carry out any experiment on that particular plant species. So understanding is most important, and understanding is possible only with the help of morphological studies. So, for example, if you choose a specific millet or even the specific plant material, then you have to find out the specific morphological characters. Because when you go for such kind of things, if you focus on specific characters, then later on it's possible to correlate a specific morphological characters with specific compound, with specific biochemical compound or specific phytochemicals. And then here number of times students, for example, if the plant material is millet, and then there is, you have to, in case of the millet, for example, you have to go for a specific character. Specific character means the yield contributing character. And for example, if the plant material is like a Pinda cucnensis or a Dicliptera or any other specific plant species, medicinal or aromatic plant species, then you have to focus on specific part. For example, Pinda cucnensis, then you have to focus on the tuber. The tubers are most important. Tuber contain most of the bioactive compounds, and the seeds are important because seeds contain the aromatic oil. So you have, in case of the plant species, you have to focus on a specific plant part which are very, very important, which are important from your experimental experiment point of view. In case of the medicinal plant or in case of the specific plant species, if you focus on the vegetative part or other characters, then these characters may not be useful to find out a specific thing or specific result. So the plant matter morphological studies, if you like to carry out morphological studies, then you have to be very specific. You have to be emphasis on the yield contributing characters in case of the crop and specific plant part in case of the plant species. When you would like to work on the bioprospecting or if you would like to work on the metabolic specific studies. So here again I will say the nutrition analysis, nutritional parameters. So try to understand the phytochemical. Number of times the students select the plant and they work on a biochemical or the nutrition analysis. And that plant, that specific plant is a medicinal plant. So here you should not emphasize the nutritional qualities. So, for example, if the plant is medicinal, then you can emphasize on the antioxidant potential of the plant. So, what I would like to tell you here that as per the plant material, you have to fix your objectives. Objectives are always based on the plant material or always based on the nature of that particular plant material, the plant species. Then again important thing is that when you when for example if you have chosen the specific 8 character 10 character morphological characters, And for example, if you collect the 100 varieties or the 100 land races for a specific crop, then I would like to tell you that it's not possible to cover the morphological, nutritional, or molecular studies of all these 100 plants within the period of 3 year or even the 3.5 year, because 100 sample, this is the big sample, you can say huge sample. It's not possible to go for all these parameters. So at every step, for example, you have 100 varieties. First of all, collect the 100 land races, 100 varieties, then screen all these 100. While screening, apply few parameters, for example yield contributing parameters, and on that basis, out of 100, choose, go for 30 to 35 or maximum 40. Again, out of 40, for example, if you had done the morphological studies, complete the studies of the 50 plants, and now the next stage is the biochemical, phytochemical, nutritional studies. Then again, I would like to tell you that it's not possible to go for all the 50 plants. If you would like to go for all these three parameters, means phytochemical, nutritional, or the other parameters, again you have to select the few uh, varieties, or the few out of 50, go for uh, 10 or 15. Means, for example, if you complete the 50 on the morphological, after again on the basis of morphological characters, try to screen elite land races, elite varieties, or the best plant material on the basis of morphological characters, and then remaining what the figure is 15 or 20, then you go for in detail. You can find out the various, you can compute the various objectives. Means you can compute the nutritional parameters or the antioxidant parameters or the phenolic compounds. And finally, again, out of this 15, 20, go for 3 or 4 plant species or 2 or 3 plant materials specifically for molecular work. Because within a short period, within 3 or 4 years, it's not possible to go for so many parameters. It's not possible to complete so many parameters within 3 or 4 years. So you have to be very specific for your parameters, and obviously the expected results. So, for example, parameters, morphological parameters, height, head, weight, these are the few parameters in case of the crops. But again, I would like to tell you that the most important parameters are the yield contributing parameter, year length, the number of grain per spikelet, number of figures, and obviously the yield per plant is most important. Uh, Biochemical parameters, again, here you have to be very specific while while performing, while working on the material methods. What exactly you would like to use? Whether you would like to use the previous methods, what are the methods are available, what we got the standard methods, or you would like to use the modified methods, because nowadays number of modified protocols are also available. So what exactly you would like to refer? For example, if you would like to determine the minerals, then Saharavat method is available, which is very popular, Saharavat ETA. But nowadays, instead of Saharavat, instead of these conventional, these available method, previous method, you can go for ICP OES method. ICP OES method will give you the exact, or you will find more accuracy with this method. Again, same thing for the antioxidant activity. Number of methods, more than 8 or more than 15 methods are available for the antioxidant. But everybody talk about DPPH. Why DPPH? Other methods are available. You can work on the other methods. and find out the most sensitive method for your crop, for your plant species, or the particular plant species. So try to, again, I'm telling you, try to understand the difference between the phytochemicals, biochemicals, the nutritional components. And as per this, you have to choose, as per the nature of the plant, you have to choose the parameters. Do not choose parameters randomly, because justification of each and every parameter is most important. And if you justify, you will understand the importance of that plant material, or you will understand how that plant material is important for the welfare of the human being, or the society, or the particular ecosystem, or the other advantages. So, as I said about the antioxidant, these are the few methods: DPPH, hydrogen peroxide method, ferric reducing method. Number of methods are available, but which method is the best method? We cannot say like this, that DPPH is the most important method, or FRAP is most important method, or most sensitive method. It depends on your plant part. It depends on the plant species. Or if you would like to work on the only antioxidant, then number of methods are available, and you have to go for all these methods and try to compare these methods. When you compare, you will realize which method is more suitable, which method is most sensitive, and this is also the part of your experiment. Then, as I said about the modern tools, a number of modern tools are available. For example, LCMS analysis. uh, why to go for, first of all, when you think for the specific modern tool, then ask question: is there a need really this LCMS, or a specific modern HPLC or HPTLC or MSMS? This is really the required, is the requirement of your work. If the answer is yes, then you have to work what is the LCMS, for what the LCMS is used, then what kind of compound you would like to use for this LCMS system. So what are your expectation exactly? So here means, for example, LCMS, then here, for example, phenolic or polyphenolic compound, then quantitative study and qualitative study. And here I would like to tell you that the hiring charges are more charges, very huge charges. If you have 20 sample, 15 sample, it's not afford if you don't have funds. So quantitative study and qualitative study. Okay, generally, student, when they would like to utilize this LCMS, they generally talk about the qualitative study only, means the name of the compounds. But if you would like to beyond that, if you would like to go for the quantitative, then you will get more precise, the specific name of the polyphenol, monophenolic compounds, fractions, all these things, and in how much amount these compounds are available. So quantitative and qualitative both are important, but depending on the funds, you have to choose a specific areas, whether qualitative or quantitative. Then same thing is for the amino acids. These are the few modern tools. I'm not going to explain the each and every tool or its importance. You can go for the LCMS, HPTLC, GBS. I'll talk about the GBS. Then UV, FTR, molecular marker, same time protein profile, etc. But again, I am telling you, before, when you prepare the blueprint of your research, then you have to find out various angles. You have to work on various angles. As I said, the FTIR. Why to go for FTIR? Then what kind of compounds? What studies you would like to undertake? So these things are most important. Same thing is for a molecular marker. Number of markers are available, but what are your objectives? So these things are most important. At the same time, again, I am telling you, so fix your objective, fix your parameters, and finally, the most important thing, the hiring services and their charges. So, for example, if you would like to go for this genotyping, GBS, genotyping by sequencing, molecular sequencing, and all these things, then obviously here the sample preparation is the important part. In the beginning also I emphasize on the sample preparation. Everything means entire experiment is based on the sample preparation. So sample preparation and the material, plant material, whether the seed, leaf, whatever things you would like to use, so that material is most important. And you should understand about you should understand the plant material, everything about the nature of the plant and the sample preparation. Then obviously here you can see the few important things: sample preparation, then library assembly, means poor samples, size selection, quantification, then adjustment of the concentration, then sequencing, then this SNP calling, and find for what all these things is here. If you would like to go for this point, if you would like to find out, work on the diversity analysis of diversity, then this GBS is most important, and it will gives you idea about the heterozygosity, genetic distance, genetic relationship, genetic differentiation, and genetic structure. Again, I would like to tell you that it's not possible. Even you can emphasize on only two parameters. You can only focus on the heterozygosity, or you can only consider the genetic structure. So even it's means out of 4 5 parameters, you can focus on a specific parameter, and as per the parameter, you have to choose a particular method. Then the most important part, results. So while writing the result, this is the most important part, because we when we talk about the material and methods, this is a little bit easiest section, because simply you are reproducing the material, does not require a lot of imagination. Most of the literature is available, so by using that literature you can complete the result, you can complete the material and method, this method part, very easily. But when we talk about the results, then here you have to you definitely remember, here more efforts are needed, lot of efforts needed to complete this part, because this result include the interpretation. So first important thing is that you have to organize the obtained data, what your data you will get. First of all, the organize the data, then put your data in specific form, in table, graphs, and all these things. Then try to interpret your result logically. This is most important. Interpretation of your result logically means what exactly? Suppose if you check a specific plant material for the calcium content, and calcium content, as per the available literature, calcium content is suppose 9.2 milligram per 100 gram. I'll repeat once again, 9.2 milligram per 100 gram. But in your plant material, if you in your specific plant variety or the land race, instead of this figure, if you get 25 milligram per 100 gram, then this is the big gap. Logically, this is not acceptable. Means double the, for example, calcium, double the amount of calcium. Is it possible in nature? Or for example, 10 milligram per 100 gram, if you get the 25, 30, 40 milligram per gram, then you have to check, you have to Again, I am telling you that the references are most important. Find out each and every paper which deals with the calcium content of that particular plant material, because 10 milligram per 100 gram as per the available literature, and as per your result 30 milligram per 100 gram. So huge gap here you will find. Might be the things went wrong during calculation, dilution, and number of things are there, or maybe it's a miracle, maybe it's a surprising result. So do not avoid these things. Repeat the experiment number of times, 2 3 times. If you get a 30 milligram per 100 gram, because it's beyond logic, then perform in your laboratory, then at the same time send sample to specific institute or private laboratories, government laboratories, to check, to compare the results. And really at the end, if the amount is 30 milligram, then definitely remember, this will be the great findings. But here in the beginning, you have to use the logic. Number of times 10, 20, 30 milligram. In nature you may not find such kind of things. If the material carries 10 milligram, then you may get 12, 13, 15 milligram. Okay, but 30 milligram is not possible. Okay, I'm not saying not possible, it may possible sometimes, but 99% it's not possible in nature. So second important point is that focus on the main findings of the result as per the objectives. Obviously, what are the main findings of your results? You must clearly explain what did you find. So what did you find is the most important, but at the same time, one more thing is most important, that what did you not find. So you have to write the both, you have to focus on both. What did you find and what did you not find? Then the most important thing is that you must be very transparent while writing the result, and need to provide all details, all the required details. Number of times we hide a few details about that particular data, particular experiment. So absolute transparency is most important. Then one more important thing is that number of times we obtain unexpected findings. Again, I will say that this is again important part. Unexpected findings are most important, and this is the because you are pursuing research, you are performing some kind of experiment, and you are expecting specific things. But while doing so, you may find few unexpected results, and these results are most important. You have to explain very clearly whether the obtained results are significant or not. Means again you have to use the different tools, and by using that tools you have to clear whether the obtained figure, obtained values, they are significant or not. Again, here I would like to tell you two more important things, that in the result part, emphasize, give maximum emphasis on the figure and tables. When to go for figure? If you would like to show the trends and pattern, decreasing trend, increasing trend, or the pattern, variation in the pattern of the growth. So means when you would like to show the trend and pattern, then definitely we have to go for the figure. you should not that time you should not use the tables. And when you want to show actual values or the numerical values, lot of numericals, then that time tables are most important. Number of times when confused what to use exactly, whether tables or graph. So as I said, so it depends on the data. If you would like to go for numerical, for a lot of numericals, then always tables. And if you would like to go for the patterns, trends, and all these things, then the figures. Again, as I said, the significant or not. So need to discuss the obtained result here. Then discussion part is the most important. In case of the discussions, first of all,